Good afternoon. On behalf of the Iziko Museums of South Africa, welcome to the final day of the second Iziko Virtual Symposium. My name is Glynn Alard. I'm an educator based at the Iziko South African Museum, and I'll be facilitating the proceedings for today. The Iziko Virtual Symposium forms part of the International Museum Day public program. The year's theme, 2021 for International Museum Day, is the future of museums, recover and reimagine. Like all institutions throughout the world, museums have been affected by the COVID-19 pandemic. Through impacts and our operations, we have experienced a drop in visitor numbers and our operations have been affected. In trying to recover from the fallout of the current pandemic and to ensure our survival, museums have to reimagine a new way of operating in a new world. One of the continue connecting with you, the public, is through virtual platforms. And so like the symposium, Iziko Museums have thrown open our virtual doors, allowing the public to view online exhibitions and public programs, such as this symposium. The symposium allows you to personally connect to Iziko experts, and this is not usually the case when you have a physical visit to a museum. We hope that you will make use of this opportunity by sending us your comments and your questions through the chat function in Zoom and also through commenting on the Facebook Live comment section. We thank you for joining us today and for your support and interest. Please continue to follow us on our website and all our social media channels such as Twitter, Facebook, and Instagram. The session will be recorded and your audio and video functions will be disabled. The question and answer discussion will be held at the end after all three presentations have been delivered. Please type and submit all your questions and comments to me in the chat function and type the relevant speaker's name so that I can direct the questions to them. If you have a look at the bottom, if you enable your chat function, you'll see that there's quite a few options. There's a drop-down list. The top one should be everyone. If you find me, Glynn Alard, please try and direct these questions for my attention so that I can direct the questions appropriately. We anticipate that we'll receive many more questions or comments than we have time to address. So we do apologize if we don't get to your question or to discuss your comment. Please make sure that your video has been turned off and that you select the viewer setting. The virtual symposium has been running for 3 days. Today is the final one. And Iziko has three main focus areas, which is art, social history, and natural history. We had very interesting and lively discussion around art on the first day, yesterday social history, and today we present to our natural history team. Our first speaker for the day is Aabid Abrams, and he is a research assistant intern at Iziko Museums of South Africa. His job entails processing field samples and data, and the digitization and curation of specimens, scientific technique that he applies in his job. He obtained a BSc in Biodiversity and Conservation Biology at UWC after nurturing a lifelong interest in the natural world. He's definitely, of the entire symposium, one of the most enthusiastic and passionate presenters, and we really do hope that you enjoy his presentation. Aabid, over to you.
Hi everyone, good day. My name is Aabid Abrams and I'm going to take you through a day in the life of a research assistant. I'm a research assistant intern at Iziko Museum of South Africa. My job entitles processing field samples and data and digitization and curation of specimens on a day-to-day basis. I process field samples by sorting insect species to family level, as well as counting those specimens. Once species are sorted and separated into family level, data is put onto an Excel spreadsheet, and specimens are put into a soda glass flat bottom tube with respective labels. before it is stored in the wet collection facility. Digitization of specimens is done on a software called Specify. This allows data to be stored on a system that is organized and reliable for future use. Why is my job important? The importance of this job is to ensure the effectiveness and efficiency of the collection, as well as protect the credibility and reliability of the data. How may people help? People could increase their insect knowledge by visiting the museum. They would learn some of the factors that have an influence on the insect life cycle and their distribution. Creating awareness is very important. Example, urbanization has an impact on the environment. This affects the natural species as well as the use of dangerous pesticides such as dooms and garden fertilizers. Having individuals that are aware would reduce species extinction. Interesting facts. The word insect derived from the Latin word insectum, which means to divide into segments. An insect body is divided into three regions, namely the head, thorax, and abdomen. Having 2 pairs of wings depending and 3 pairs of legs in the thoracic region, insects occupy 2/3 of the total population of the phylum Arthropoda. In image 3, you can see a lot of drawings. Those drawings at the beginning were for myself to identify species. Identify these species on a daily basis through wings, body characteristic, and abdomen characteristic. In image 4, there's an illustration of the insect orders that I sort on a daily basis. This is besides the Hymenoptera order. Interesting facts. There are over 900,000 insect species that have been named, of which 10,000 species are incriminated as pests of agriculture, forest, and livestock. The rest are either beneficial or indifferent. Image 5, 6, 7, and 8. What it actually looks like from the eye's view compared to image 7 and 8 as the magnification gets adjusted respectively. Image 5 and 6 looks like a mess. Once adjusted, a variety of insects can be seen. In image 7, they are all in one family called Braconidae, but look very different due to their variations in their body characteristic, all different to adapt to their specific environment or niche. Insects are beneficial, either medicinal, medical, they are pollinators, they can be used as biocontrol agents, they can be used as production of products such as honey, wax, and silk, and they are nutrient recyclers. They feed on decaying material, which improves soil structure and texture. And last but not least, they are source of food. Yummy. Crickets, grasshoppers, beetles, and insect larvae are used on a daily basis in the eastern countries as a staple food source. In this image, you can see the equipment that I use on a daily basis. I'll go through them from A to K. A metal pinning block. B, my newtons. C, a fine liner pen, specifically used on labels because the alcohol, this ink prevents the alcohol from coming out of the labels. D, a tweezer. E, alcohol bottle. F, pins. G, a soda flat bottom glass. H, a circular pinning block. I, scissors. J, a petri dish. And K, a label. Image A, B, C, and D represents my workstation. Sorting out insects to order level, such as Lepidoptera, Coleoptera, Psychoptera, and Plecoptera. Image D is the field samples that I get from the field that Simon brings back. And image C and B, you can see in the petri dishes, it just looks black basically, but those are actually flies which belong to the order Diptera, and they are moths and butterflies which belong to the order Lepidoptera. That might look like just a mess, but there is actually about 600 flies in that one petri dish and about 200 moths in the one petri dish. And I think in the smaller one, we have about there in the corner, the very small dark petri dish. I can assume that is just Coleoptera, that is beetles, that is very small beetles, and there's about 300 in there. So, and that is tiring, quite tiring to count, because that's how I have to know. I know these numbers, would have to count these. So, yeah, and oh yeah, these dishes that I use, these trays, these circular under the petri dish, those are used as for my family level for Hymenoptera. For example, family in Hymenoptera would be Ichneumonids, Crabonids, Evanidia, Chalcids, and there's about, I think, 6, 12, 18, 24 families so far that I know, and that I have to take out of these sample jars. And then the rest would be about 10 different other orders when it comes to besides Hymenoptera. So, yeah, and I'm very happy with this microscope. Yeah, it's a pretty good magnification compared to the first one I used. I couldn't get that high magnification and I missed a lot of chalcids. They're very, very small wasps. I think they less than 0.5 micrometer. That's very small. Image E, F, G, and H. Image E, the soda glass flat bottom with labels. The first label represents the family name. The second label has detailed site information which includes coordinates, dates, vegetation type, elevation, type of trap, and of course the collector. The vials with the green caps are used as the same purpose as the soda glass flat bottom tubes. Image H has a catalog book. The catalog numbers, once separated and identified to family or species level, they are given a catalog number and put onto a software called Specify. This helps document and keep a record of species kept in the wet collection and dry collection. Finally, color. Yeah, this makes me smile. I don't know. Yeah. As you can see in the previous slides, just black and white. And these images represent what I see through my microscope and the beauty. So we have I, J, K, and L. In I, we have a Coleoptera with beautiful metallic body. These are the colors and beauty that I see through the microscope on a daily basis. J looks like an ant, but trust me, it's not. It's actually a wasp. K, the most beautiful of them all, the gem wasp. And L looks like an ant, but actually really is a spider mimic. Wow. The image above is from 2019 Heritage Day. The Entomology Department had an exhibition for the public. Edible insects were served, such as mealworms that had been caramelized and party worms that had been salted and dried. On this day, the light is coming there, but they grabbed hands full. We're talking about hands full. And then they were eating it, and we told them it's worms, and they pulled their face and they just looked, and then they went on because it's so sugary and nice, no? That they, yeah, it was really funny. I enjoyed it. They didn't worry about the pani worms because it was too salty, but the mealworms is number one. So it can be used as a staple diet, you know. Can feed probably school kids on a daily basis that don't have, you know, swap the sweets instead. So save the teeth from rotting as well. But yeah, then I still have something to say when I want to end off, like for a fun fact. But I don't know if it's gonna offend anyone, so may I say it? But the fun fact: if a pair of houseflies is left to breed from April to August, they would produce progeny enough to cover the earth in a layer of 14 meters. Such a case will only happen if a fly remains alive and produces continuously. Another fun fact: cicadas have a lifespan of 17 years. The female member of cicadas is voiceless. So there is a saying, happy the cicada's life because they have a voiceless wife. Thank you.
Aabid, thank you very, very much for your interesting presentation. Aabid was very, very nervous, but that was absolutely fantastic. Thank you. Our next speaker is Sibusiso Mtungata. He is a fossil preparator, and I was privileged enough to go out with the Karoo Paleontology team to the Karoo last year. And he truly is one of the most talented field technicians that I've ever seen in action. So I'm particularly interested in this presentation. He's been a professional museum technician for more than 10 years, and he works as a fossil preparator in the Iziko Karoo Paleontology unit. He's an expert fossil finder and has formed part of the fieldwork teams in South Africa and Namibia on research teams in the Permian time. This is roughly 250 million years ago. In the fossil preparation lab, Sibusiso is a specialist at removing the rock from delicate small fossils, concentrating on tiny skulls and teeth. His meticulous work has made many scientific discoveries possible. And in 2021, he was justly rewarded for his contribution as a co-author on a scientific paper describing the latest research on the biggest mass extinction event the world has ever seen. Sibusiso, we look forward to your presentation.
Thank you for having me here. My name is Sibusiso Mtungata. I am a museum technician working as a fossil preparator in Iziko Karoo Paleontology unit. This is a typical Karoo scene. The Karoo Basin is one of the most remote areas and spectacular, as you can see from the image there. It is one of the places that we normally go to do our field work. It is one of the most important places in the world because it is yielding long past life. And the view there this is a central Karoo area, a small town called New Bethesda in the Eastern Cape near Graaf Reinet. you can see there's nice, beautiful mountains and hills, and the smaller mountains are just covered with mudstones and sandstones. And at the back there, you can see the campus back, and it's quite resistant to erosion and weathering, only because it is kept by a non-weathering dolorite. This is a campsite. This is our home for a few weeks. We're out there in the field. At one stage, we once slept on the scalp crawl because it was dark, and then we couldn't find our way in the dark because we had a long drive from Cape Town to the Karoo. So knowing the Karoo of lacking of limited cell phone reception and signal, so it's always best just to find a site that you can sleep overnight before you can start a day and look for the site that you're looking for. But because we've been doing field work for quite some time, so it's quite easy for us to adapt to this when we meet these such circumstances. Finding and looking for fossils. When you find a fossil, it is a jump of joy moment. But before celebrating, there are other steps that you have to follow. You mark every occurrence with a removable marker or biodegradable marker like toilet paper, and you recover every loose bones. When you find the fossil, you have to look around for any other loose bones that belong to the fossil that you have found before you can sit down and do any other thing and do all the recordings. It can be quite difficult sometimes when you don't have trained eyes to find fossils. As you can see there in the picture there, this is a Lystrosaurus skull with a steep snout, and what is quite visible, and the orbits and the nostrils, you can see them. If I can just point there on the image, but it's not that easy. At least you must have a day or maybe some days to have your eyes get used to the environment. You can see that the color of the fossil is very similar to the color of the environment. Excavating. Okay, when you do excavating, especially disarticulated fossils, you have to look around for any other loose bones that might belong to the same fossil before you can start any other thing. And when you do excavation, we use rock drills, hammers, and chisels, and it might take a few days before you can uncover the actual fossil. And once you manage to do the uncovering, like we call it undercutting, when we have managed to take out the fossil out of the ground, we use a black bag, which acts as a separator between the fossil and the plaster jacket. Once we've done that, then we use plaster of Paris to make a jacket and lift the fossil and add on a field number to the actual fossil once we're done. As you can see there, this image of me using a rock drill can be quite difficult and you need to be very careful when you use this. And you can see I've used all the suitable personal protective equipment, because it's noisy, it's dusty, and it's very dangerous because it's got a spinning blade. When we do transport fossils from the slope to the vehicle, it can be very tricky because of the unevenness of the ground, the weight of the fossil, and the thorny shrubs of the karoo. So it is very important to have teamwork so that we don't get hurt. And the other thing is that sometimes we do require hired help because of the weight of the fossils. Here we are seeing a fossil being hoisted at the museum, and it's going to be taken to the prep lab and then be prepared. And what happens now is that after we go to the museum, sometimes we bring monsters as big and heavy as a ton to the museum, and once they get to the museum, we have to open them with open the plaster jacket, and then if there's a need for the reconsolidate, we use thin glue. Then once everything is done, then we just start preparing. And it took about 2 years to prepare this monster you see in the peak here. But for other smaller ones, smaller fossils takes about less than that time, maybe 2 weeks or a month. Fossil preparation is done using compressed air scribes, and magnification to reveal the taphonomic details of the bone surface, and such as weathering stage of the bone, and tooth marks, and skin impression. But in general, it's just to uncover the whole fossilized stone bone. And when we do fossil preparation, we observe all the safety rules. As you can see there, we use suitable personal protective gears, and especially from the dust that's coming out of the rock, and with the buzz, sound, and the vibrations that are coming out of the scribe, the vibrations are not that harmful. Just vibrations, but the sound, the buzz sound, we use suitable earmuffs just to protect your ears, and we put on our lab coats to protect from the dust of the rock. The demonstration lab in the exhibition area at ISAM, the South African Museum, is one of the places that we normally use to interact with the public and the school groups. It is quite very important to engage with the public and sharing of knowledge and skills that we have obtained. But it can be quite very tricky at times, because when you have to talk and explain about the past life, long past life evidence, that is difficult to comprehend, and the extinction of animals that we can't even relate to, and special evolution, that's a very, very tricky subject to go to. but is something that we enjoy just to exchange ideas and arguments and debate about all of this stuff. Thank you for allowing me this time, and further thanks to the team of Karoo Paleontology team for tolerating my imperfections, especially out there in the field.
Thank you very much, Sibusiso. We have a very strong representation of entomology today, or the study of insects. And our final presentation for today and for the virtual symposium is Dr. Terry Reynolds-Berry, who is a postdoctoral fellow at the Iziko South African Museum. She obtained her PhD in zoology from Stellenbosch University under the primary supervision of Iziko's curator of entomology, Dr. Simon van Noort. Her research focuses on the systematics of parasitoid wasps, particularly within the subfamily Bankinae from the Afrotropical region. Terry, take it away.
Hi, my name is Dr. Terry Reynolds-Berry, and I'm going to talk to you a little bit about creepy crawlers, who, how, and why we collect them at the Iziko Museums of South Africa. Iziko houses the largest entomology collection in South Africa, with specimens dating as far back as the 1840s. The collection includes spiders, scorpions, insects, millipedes and centipedes, springtails, mites, and ticks. And the collection is impressive, with more than 1 million pinned insects and even more preserved in alcohol. Just the other day, our curator said that he collected about 100,000 new specimens on one of his sampling expeditions, which goes to show that the museum's collection is ever growing. And with such a large collection, it requires a lot of maintenance and manpower, expertise and dedication. And so a lot happens behind the scenes of the museum that the public don't know about. And so let me introduce you to the team. The entomology team consists of 3 permanent staff and 4 contract staff. We have 2 curators, Dr. Simon van Noort, who is a leading research scientist on African Hymenoptera, and PhD candidate Nokutula Nhleko, whose research focus is on Southern African ants. Their work is made easy by the help of our collections manager, Aisha Mayakiso, who oversees and does inventory of the physical care of all the specimens in the museum. When specimens come from the field, every specimen is handled by our research assistant and collections assistants. And we have a postdoc, which is me. Under the mentorship of Dr. Simon van Noort, my research focus is on parasitoid wasps. There are an array of collecting methods in which samples are collected in the field. And the reason for this is that we want to collect as much of the diversity in that area as possible, because certain insects are active during the day and they are attracted by bright colors, while other insects are active at night and they are attracted by a light source. Some live in leaf litter, while others make nests underground. Some are flying insects and others are not. And so we have all the different collecting methods so that we can cover our bases. My role as an assistant curator at the museum is, after the research assistant has sorted all the insects into their different families, I then pick out the family that I work on and sort it further down to subfamily, genus, and species level. These specimens are then pin-mounted and studied under the microscope. With the group that I work on, I'm also responsible for identifying gaps in the collection, addressing problems with classification, and updating that information on the museum's database. Imaging is taken of the specimen by means of a multi-focus stacking system. And during photo processing, that image is made sharper and it will highlight diagnostic characters, which will then be used for species identification, which would then lead to the end result of a lovely species identification key. With the group that I'm working on, I'm responsible for identifying gaps in the collection, addressing problems with classification, and updating that information on the museum's database. It must be there now. As a research assistant, I study within the field of systematics, which looks at the taxonomy and evolution of a group of organisms, which in my case are parasitoid wasps. Investigating the relationships among species over time and space. And us researchers have all the fun of getting to discover new species, describing them and naming them. The first wasp I ever got to describe, I named after my father, Cryptopimplanelli. As a research scientist, I'm also responsible for communicating my research findings in the form of a popular article, scientific papers, updating species web pages, and involvement in exhibitions and public participation, such as this one, the International Museum Day. So why are collections important? And this is a question that I get often. So you named a species, so what about it? Well, let me put it in perspective for you. When I was a kid, a little bit of spilled sugar would result in a counter full of ants. I would find ladybirds in the schoolyard, and a trip down to Mossel Bay would have a windscreen full of splattered insects. And this is something I don't come across anymore. In fact, last year during hard lockdown, I was so excited that I came across a ladybird that I hadn't seen in years, only to discover upon closer investigation that it was the invasive harlequin beetle, the one that is outcompeting our indigenous beetle. So the truth is, insect diversity is declining at an alarming rate, and we can even notice it in our own generation, and it's worrying. So apart from its historical value, one of the purposes of museum collections is to document the biodiversity, to have a record of it. The reason why we keep sampling is so that we can monitor the changes in that biodiversity over time and space. And the reason why we name species and describe them is so that we can understand them better. I love the group that I work on, which is parasitoid wasps, because of the potential they have to feed into agriculture and biological control. Now, caterpillars, we know, are a major, huge pest on crops and forest products. And to save the crops, usually millions of rands are spent on pesticides to get rid of that pest, but they don't just get rid of the pest, they get rid of everything else, including our beloved bees, which are food security. But parasitoid wasps do that naturally. They naturally decline the numbers of pests and kill off the pests. So they have the potential to feed into biological control, but in order for them to be effective, we need to know who is parasitizing who, and that is where taxonomists come in. Scientific research helps answer questions related to biodiversity, ecology, climate change, societal, environmental, and economic impact. And furthermore, the collections offer a reference collection for the identification of material for other projects. So I hope that I've inspired you to come and visit our collections and see what we are all up to.
Thank you so much, Terry. I'm now going to be joined by our three speakers, and I hope that we receive some questions or comments for them. Hi, all of you. Thank you so much for your presentation. Very, very hard work.
Thank you.
Yeah, for your hard work in putting together your presentations. I wanted to ask you, since this is a public forum, in some of the radio interviews I've done, and so, as you may know, the public are very interested in the work we do behind the scenes, and I'm glad that we could actually speak to you today and introduce you to the public to see what's happening behind the scenes, behind the exhibitions. How much public interaction, public contributions do we get to the collection? How often do we get insects or do we get fossils being brought to the museum?
Does anyone want to start answering that question?
I think you can.
I think I'll start answer that question. Yeah. Well, concerning insects, Simon brings in not just one jar. We're talking about a whole yellow tray. There's, how much do I count? I've make about 98 of those jars, and then he comes back and he brings all of them basically half to full. And you can imagine how many insects are in there. Like I counted one jar can have approximately, all orders, 36,000 insects in one jar. That's a full jar. And that can be minimum. There can be more, because depending on the density of the jar also and the amount of insects and the size of the insects. But yes, we can say thousands of insects come in from the field and has to be processed. So I hope that answers a part of the question.
Thanks, Aabid. Sibusiso, do members of the public actually bring fossils to the museum for the museums to look at or to try and identify? I know our teams go out and we excavate and we bring them back and we study them, but do members of the public, ever bring in specimens or fossils for us to look at, or do they donate it to our collection?
Okay, there's with fossils, there's a sort of thing that is going around that when you're out there in the field, in the Karoo, that maybe we are collecting fossils to make money or to sell them. So when people find stuff, they do bring stuff to the museum, but they are hoping to sell them to the museums. But once they get there, and then we tell them that this is a national heritage stuff, so there's nothing that gets sold in there. But when we're out there in the field, also in the farms, we do find the farm workers. They do go around these fossils and they find them, and then we just help to identify, and if it is a research, if it is a research fossil, then we can explain to them that, and then we take it to the and bring it to the museum.
Okay, thank you. Terry, we've got a question for you. You mentioned that there is an invasive beetle species. Is there a particular issue about invasive wasp species in South Africa?
Yes, thank you, Tisa, for that question. We do have problematic invasive wasp species, particularly the paper wasp. There are two different species, and both of them are from Europe, and they have been coming around and taking over the Western Cape, and overtaking the natural distribution of our indigenous species. And as we all know, it's been a pest in the home environment, and people's been stung. And we actually have a small exhibit currently in the museum that showcases those wasps and how to identify them from the African wasp. And people are more than welcome to come and learn more about it in the museum.
Terry, could you also tell us, there's been an inquiry, you spoke about the two ladybug species that you had on that one slide. So I'm not an entomologist, I know very little about invertebrates. So to an untrained eye like mine, they look quite similar. When we ask the public to look for that kind of information, what are some of the pointers we can look at between the alien and the indigenous? I mean, is it possible for a member of the public to see the difference?
Oh, yes. When I came across it last year, I actually was unaware at the time that there was an invasive beetle. But I looked in my field guide, which I understand not everybody will have a field guide, and that's how I came to know it's not the one that we normally find. The easiest way to identify the difference between the harlequin beetle and ours is they have on their head an upside down, well, depending on which way you look at an M or a W, a black marking on their head that's the shape of a M. If you're looking at from the rear end forward, or if you're looking at from the head to the rear, it's then a W. That is the key diagnostic. And then also the shape, the general shape of the body is more elongated, and the spots with our indigenous one is kind of like smudgy black spots, whereas the harlequin beetle is distinct black round spots on the body.
We've got another question about how do we find the invasive wasp? And I could add to that, how do we identify it? I mean, just as a member of the public, similar to the ladybug question.
How do you identify?
Yeah, how can we, I mean, you're speaking about the invasive wasp, how do we find it, or where do we find it? How do we identify it? How do we let someone know if someone encounters it?
Aabid, don't you know more in detail how to distinguish?
Oh, I think they have specifically the number of stripes on the abdomen. So, and usually they, how can I say, they look similar to the indigenous species, but I think our indigenous species is orange, but you do get the yellow jacket one. But I think it's got to do with an amount of number of stripes on the abdomen that can determine whether they are invasive.
Yeah, the easiest way with both the invasive ones, they're similar looking, is this bright black and yellow wasps. The two distinguishing species will either have the black antennae, or the other one will have the yellow antennae, whereas the African wasp is a dull color. It's a dull orangey color. Yes. So it's very easy to identify the invasive ones in that they're both brightly yellow and black colored. And they're the ones that make those nests often. That's why they're called paper wasps, because their nests kind of look like paper. They make it, I've seen it already at people's houses. So under the your gutters on the wall, you'd find them there.
Yeah, they usually after the plyboard or your carports, if it has wood, and then they normally stripping the wood slowly off the plyboard or the carport beams, so to make the nests. So you usually find them active around woody areas, you know, stripping the wood, and that's when you will know there is an invasive species there. And they love to make nests by the washing line. So that's a number one.
Yeah.
I see that there are many supportive messages and thanks for the interesting presentations today. I would like to add to that, to thank the three of you for your preparation, for your delivery, for the extensive amount of knowledge and enthusiasm that you have brought to the session today. I'd also like to thank the rest of the 9 speakers that have been speaking since Tuesday and yesterday. I think that this has been an incredibly informative and inspired event. There are many, many people which made this event possible. I'd like to thank the manager of public programs, Dr. Wandile Kasibe, the support of the education department, a specific mention to the director of education, Mr. Hilton Arnolds, and then especially our advancement team who has provided the platform, the marketing, and the running around and the logistics to make this big event possible. I would like to make mention of Awonke, Ellen, Melody, Ohuhle, and then also the Director of Advancement, Carol Ann Davids. The biggest thanks goes to you, our viewers, our public. Thank you for supporting us. Thank you for supporting this event. I'd like to remind you that each specimen, each object belongs to you in combination with the rest of South African citizens. This is your heritage, and I implore you to come and visit us when it is safe, subject to COVID protocols, and to come and enjoy your heritage that we are looking after and act as the custodians of for you, for the public. This is the last day of the second Iziko Virtual Symposium, and I'd like to thank you for your support and participation. I wish you a wonderful afternoon and the rest of this week. Thank you so much and good afternoon.